Health and Associate Dean of Faculty Affairs in the Graduate School of Public Health at the University of Pittsburg. He received his undergraduate degree in zoology from North Carolina State University and his Ph.D. in pharmacology from Duke University, where he also completed a postdoctoral toxicology fellowship. After an initial faculty position in clinical pharmacology at Thomas Jefferson University, he moved to New Hampshire and joined the pharmacology and toxicology faculty at Dartmouth University, where he began mechanistic studies of arsenic-promoted vascular disease with their newly formed Superfund Basic Research Program. In 2003, he joined the faculty at the University of Pittsburgh and continues investigating cellular and molecular mechanisms underlying human diseases caused by environmental exposures to metals and chronic changes in cellular redox status. He has held a number of leadership positions in the Society of Toxicology and served on scientific advisory boards for the Environmental Protection Agency and the National Research Council focused on arsenic standards and metals toxicity. The NIH has funded his research for 3 decades, and he has more than 110 publications. We are honored to have him speak to us today. His presentation is entitled Memories of Arsenic Promote Cardiovascular and Metabolic Diseases. Aaron, the screen is yours.
Okay. Can you see my screen now?
Not just yet.
Okay.
And we'll ask everyone else to mute their video to conserve the bandwidth. We've got a lot of people online today.
Does that work?
That works.
Okay, good. Thank you for the introduction, Chris. It's great to virtually see old friends. I thought I'd give you a little bit of update on what we're doing nowadays with arsenic. Basically kind of create this concept or discuss this concept of arsenic basically changing the epigenetic landscape of cells such that you wind up with a memory of the arsenic stress that carries through to pathogenic processes. And before I start, I'll kind of go back to my days when I was a graduate student and why I kind of got into toxicology and study of environmental disease. And I hated this concept that they kept on saying, this idiopathic disease. And, you know, what is idiopathic disease? Something that's supposedly of spontaneous origin. And I just never believed that things didn't happen for a reason. And kind of question how much of idiopathic disease is not really of spontaneous origin, but really comes from unseen environmental exposures. And we've been evolving this to look at how arsenic in the environment really is a pathogenic exposure and a really identifiable cause of both susceptibility and pathogenesis in metabolic diseases. And that map is a heat map of arsenic in groundwater in Pakistan, published in 2017. But I just want to use it to emphasize that arsenic is in the background because of it being the 21st most abundant element in the Earth's crust. And we have been drilling wells into that crust for many years and caused quite a bit of exposure. It was estimated that with this heat map, you have somewhere on the order of 60 million Pakistanis who are drinking water that's above the EPA limit of 10 parts per billion. But I'd like to take a step back, and if you remember your history lessons, this is one of the oldest cradles of civilization. So how many thousands of years of exposure has been causing cardiovascular and metabolic diseases in this population when we really had no idea what was there? And of course, I always have to have this disclaimer that I'm a bad toxicologist in some ways because I don't like to outright kill things, and also put into perspective what we're discussing as far as chronic levels of arsenic exposure that cause disease. It's not the immediate acute concentrations, and the insidious nature of arsenic is that in the glass on your right, I couldn't tell you whether there was absolutely no arsenic or a milligram of arsenic in that glass of water because it is odorless, colorless, and tasteless. But what we're mostly interested in is lower to moderate levels of exposure that may be driving chronic changes in disease patterns. And my work has been a little bit distinguished from a lot of others in the field of arsenic in that I have primarily focused on cardiovascular disease and now metabolic disease a little bit more. But why cardiovascular disease? Essentially, if you look at cardiovascular disease and perhaps lung disease, they're the major killers of non-cancer type diseases caused by arsenic. And this was some work we did with Anna Navas-Asen and a number of other epidemiologists, really trying to nail down the dose response for environmental exposures causing cardiovascular disease. You see so many authors because this was a really unique effort in which each of those authors shared their large database of prospective epidemiology studies, such that this dose response analysis was done with upwards of about 250,000 individuals throughout the world. And what we found was that basically, even at the EPA drinking water level of 10 parts per billion, that you have an inflection point for increased risk. And so it says two things. One, yes, there is quite a bit of incidence and mortality caused by arsenic with cardiovascular disease, but also that the water standard that's based on the cancer risk may not be protective of all diseases or may not give you much of a margin of protection for various diseases. And Chris and I have been on the National Research Council Committee on Inorganic Arsenic, and this has been a big issue with EPA reevaluating the current standard. So many of you are familiar with much of our work over the years with cells and with mice, and many have worked with mouse models. We even had a wonderful meeting, Chris, I guess that was about a decade or so ago, where there was a number of investigators who got together and talked about models of how to study arsenic in mice. And so basically, we've just been putting arsenic in the water, letting the mice drink it, and then evaluating their health and the health of their tissues after various periods of time. And this is a figure that summarizes basically all the signal transduction discoveries we made from 1994 through 2017. And essentially, our work was just defining how arsenic interacted with cells to activate transcriptional programs that led to vascular remodeling, tissue remodeling in a pathogenic manner, also with angiogenesis. And noting that we have a range of different signaling events that we could ascribe to activation of specific reactive oxygens, species generation, but then we also have general stress responses. And depending upon dose, you have a balance of which ones of these are fairly dominant. So we moved away from the straight assessment of how arsenic changes blood vessels and moved a little bit more towards how it remodels various tissues. We've looked at the liver, we've looked in adipose tissues. And the work that we've been doing for about the last decade is focused on muscle. And why muscle? Essentially because muscle is the largest metabolic organ in the body. And when we disrupt muscle metabolism, especially insulin sensitivity, it's a major driver of cardiovascular and metabolic diseases. We see compositional decline in muscle over aging with a increased infiltration of fat in the muscle. And this is actually fairly predictive of the risk for all-cause cardiovascular mortality as well as type 2 diabetes. And really, insulin resistance is a major driver of these changes and changes in glucose homeostasis that promotes the cardiovascular and metabolic disease. And of course, we know that the population is aging quite quickly, and there is a greater increase in muscle weakness being a great factor in contributing to the decline of essentially healthy aging and a strong predictor of mortality. And yes, it has been shown in epidemiological studies that arsenic has an effect on muscle metabolism, muscle atrophy, and then also another dimension is that it will affect the sensory motor operation of muscle. And this has actually even been seen in children in Bangladesh who are only 10 years old. So speaks to whole life exposure impacts on muscle and that may likely lead to disease. We also were fortunate enough to gain a great collaboration with Dr. Fabrizia Ambrosio at Pitt, and she came to me about a decade ago and asked a simple question. She's very much involved in investigating tissue regeneration, muscle rehabilitation, and asked this question of if you were exposed to a contaminant in the environment and you were injured, let's say, and you're 30 years old, would you repair that muscle and regain function like a 30-year-old, or would you have the regenerative capacity of an elderly individual? And we developed a great collaboration with some great R01s and evolved this into asking the question of whether or not arsenic promotes changes in muscle metabolism that will contribute to metabolic disease, impair regeneration, and increase mortality risk. So 2014, we published that yes, indeed, arsenic impairs the physiologic function of skeletal muscle. We did this in vivo. In this case, we had a 100 microgram per liter, 100 parts per billion arsenite exposure in male mice for 5 weeks, and then tested the physiology of their muscles. Specifically, we were looking at the function of the tibialis anterior muscle and its performance, strength, as well as fatigability. And it turned out that in an uninjured muscle that from mice that were drinking arsenic, the main thing that we saw was that the muscles would fatigue faster, and then they would not recover from fatigue well, indicating that they had really shifted their metabolism quite a bit, lost a good bit of oxphos metabolism, and had consequence. But the other major thing that we saw in those muscles was that metabolic shift was likely due to profound effects on the mitochondria in the muscle. So these are TEM of muscles from those mice. You can see, see if I get my pointer. You can see really nice, healthy mitochondria here in these muscles, good formation of the muscle bundles. This is just a larger magnification of this area. But in the arsenic muscles, a little bit of loss of definition of the muscle bundles, but really profound remodeling of the mitochondria with a large amount of vascularization and certainly loss of health. We could do whole fiber oxygen consumption, and we found that they actually had increased oxygen consumption in the arsenic muscles, but it did not seem to be functional. It's probably more that associated with an increase in reactive oxygen generation. We also saw that there were gene changes in muscle remodeling enzymes, and this could account for some of the loss of these cristae that you see in the mitochondria. So the other thing that we found in the same mice were that we were looking at adipose tissue, white adipose tissue, and we saw changes in that adipose tissue. But then we looked in the muscle and we found that we had an increase in ectopic deposition of fat. And this is actually a earlier indicator of insulin resistance than you would see with a change in glucose levels in the blood. But it was somewhat dose dependent. We had some other indications that if we went out to 10 weeks, say, or longer, we could see the same perivascular interfiber gain of fat in the muscle. And in addition, we used an injury model to test what happens with regeneration. And in this model, we stop the arsenic. We injure the tibialis anterior muscle by injecting cardiotoxin, and then allow those muscles to recover in the absence of arsenic for 2 weeks, and then evaluate them. And what we see, and histologically, we see that we have a healthy regenerating fiber here in the control, but much fewer regenerating fibers, more cellular infiltration, and a poorly regenerating fiber with the arsenic exposures. We can quantify that. In this case, we were looking at myofiber diameter, and you can see, again, you just don't have as many nice fibers coming back. And we also can use sophisticated imaging with second harmonic generated multiphoton confocal with 3D reconstruction. And here you're looking at collagens. Here you're looking at autofluorescence of the fibers. And it's easy to see in many of the different images that you really just do not regain good fibers in the mice that had been exposed to arsenic. And we could also do our physiology measurements. In this case, we waited 4 weeks, and still, even after 4 weeks, the muscles had not repaired, and they had lost a lot of their ability to generate force and strength. So interesting global observations, but what was driving a lot of this? And we came back again to those mitochondria and mitochondria remodeling. In this case, these were mice at 2 weeks, 5 weeks, isolated their muscles and did TM again. And you can see that what we usually see is these large networks of mitochondria as opposed to nice rounded fragmented mitochondria. And then also as we progress, we see these large mitochondria, but then we also see a lot of large lysosomes and autophagal lysosomes. This has been described by others as frustrated autophagy and certainly not healthy for the muscles. We also could take the muscle stem cells, the satellite cells, out of these muscles and put them in culture and look at their oxygen consumption. And what we really found was somewhat striking and confusing at first, but we could take these cells out of the mice, culture them for several generations in the absence of arsenic, and we still retain this aberrant structure of the mitochondria, enhanced oxygen consumption, enhanced ROS production, and enhanced autophagy and poor differentiation. And this is where we somewhat came up with this concept of a memory of arsenic exposure and curiosity about how that memory was generated. So to dive into that more and to show you why we were intrigued by this, these are muscle stem cells, satellite cells, again, isolated from either control or arsenic exposed mice, put into culture for a period of time, several generations in culture without arsenic, and then looking at reactive oxygen generation. This is just the mitochondria, and you can see that there's a much larger mitochondrial network in the arsenic cells from the arsenic mice. A larger amount of reactive oxygen species generation. We can certainly quantify this, and we see that we just have a chronic elevation of ROS in the arsenic exposed cells. And what we also were just amazed with was we tested the oxygen resistance or the ROS resistance of these cells by hitting them with a nuclear bomb of 200 micromolar hydrogen peroxide and then seeing what happened. And you see the control cells did what you'd expect them to do. They basically died, stopped proliferating, whereas the arsenic exposed cells basically went dormant for a period of time, but then started proliferating again, and indicating that they had gained a dysfunctional ability to resist reactive oxygen species. So we went to a ideal cell model, the C2C12 myocyte model, to investigate mechanisms a little bit further here. And this is a publication from 2019 where we use this model to essentially look at the ability of the myoblasts to self-renewal and their behavior over a replicated age. This is the healthy condition. What we found with arsenic, again, changes in the mitochondrial structure in some of the cells, in the populations of the cell. And we saw that we had maladaptive self-renewal. And I'll go through some of this data to show you that we think that this maladaptive self-renewal is really driven by major changes in the mitochondria, speaking differently to the nucleus, and how we reverse all this. So the simple model, we put the myoblasts in culture with 20 nanomolar arsenite. We've shown this is a dose that they're affected by, but certainly is well below any kind of toxicity. And it will affect their differentiation. We allow them to differentiate over 4 days. Then we harvest the cells, put them back into culture, but without arsenic. And then sometimes we include compounds that affect the mitochondria. And then we look at the progeny of these cells. So they've seen arsenic here, but no arsenic here. And we're looking at multiple generations after we removed arsenic for a memory of that arsenic response. Just a dose response in this model for differentiation. The green is Desmond positive structures, myotubes that are formed. We can quantify the number of fused structures compared to individual nuclei on differentiated cells. And you can see that we start losing the ability to differentiate somewhere between 20 and 50 nanomolar in this model. We can also look at fiber length, and you can see, again, we're starting to have effects in this range. And yes, we can, again, look at the ratio of those undifferentiated cells, which we call reserve cells, versus the formation of the myotubes. And you can see that arsenic greatly increases these reserve cells at the expense of tubes. And what's happening here with differentiation is that arsenic is stimulating the proliferation of the cells, but not their differentiation. And we can look at that in a number of different ways. CFSC is a dye that stains the cells, and as you divide, you dilute the dye. So we can see that multiple generations, there's more multiple generations with the arsenic. Ki67, increase in cyclin. We've shown that in many different models that arsenic basically leaves cyclin D on when it should be cycling on and off. And looking at stem parameters such as CD34, we see that we have an increased abundance of CD34 positive cells. Those are proliferative stem cells. And this is also in cells that are increasing in their mitochondrial area, looking this with MitoTracker deep red staining. And again, this is population doublings, and the cells that are more doubling will give you this greater mitochondrial network. Looking at mitochondrial function, it's really easy to kill the mitochondria with arsenic, depending upon dose. But in our case, we're not killing the mitochondria, as opposed to something like an uncoupler like CCCP that causes a depolarization and apoptosis. We actually see a hyperpolarization or an increased polarization in the positive direction with arsenic compared to control. We have, as I showed you before in the satellite cells from the arsenic-exposed mice, an increase in ROS production. We have an increase in mitochondrial mass, and you can also see that we've got these long structure of mitochondria with, again, that same Christie remodeling that you saw in vivo. And we have the same increased changes in the Christie remodeling enzymes such as OPA1. It's a multi splice variant, so you see many different bands, but overall we have an increase in OPA1, YMEL1, and OMA1, all remodeling enzymes. Cardiolipin is the major lipid in the respiratory chain and in the inner mitochondrial membranes. And this dye, non-acridine orange, will be green if it's in a reduced form. If it gets oxidized, or it will start moving into the blue area. So basically, we're seeing more oxidized cardiolipins in the cell. In response to that, you also see an increase in cardiolipin synthesis or synthase. So a lot of extensive remodeling of the membranes and gaining a large network. But this is not oxygen stress or cell death. If you define oxygen stress as when oxidants are generated to an extent that it overwhelms the internal ability to reduce the oxidants. An indication of this is that we don't lose glutathione. We never lose glutathione. I published this in 1994, 5 or so, that arsenic at these low levels only increases glutathione, most likely through increasing NRF2 signaling. And if you look at something like caspase activation for apoptosis, strikingly, we actually do see an increase in caspase activity. And so this is CFSC, so population doublings versus caspase. And we can see an increase here in the arsenic-exposed cells. But if we look at annexin staining for apoptosis, we actually don't see any change at all. So this is actually processing other than destroying the mitochondria and causing apoptosis. So we're well below any kind of cytotoxicity, but we're certainly changing the phenotypes of the cells. Why do we think that this is really connected to the mitochondria? And this also brings up what we can do about it. We've used compounds such as XJB5-131, which was developed as a mitochondrial-directed scavenger of radicals, superoxide. This was developed by Dr. Peter Whiff at University of Pittsburg. And this is just to show you that this is really an interesting molecule that's catalytic and can really reduce the amount of electrons that might be going to superoxide in Ross generation. Same model, and we're taking away the arsenic here, plating the cells, adding XJB, and then looking what happens. So this isn't preventing arsenic from causing changes. It's reversing the changes after we've removed arsenic. And what we see, again, membrane potential was increased in the arsenic prime cells, but it is definitely reduced with the XJB. And same thing with mitosox or Ross generation, same thing with the mitochondrial mass, that we can reverse the effects of arsenic on the cells by adding this compound. And we also can reverse that effect on proliferation, here measured by CD34 or CD133 markers for stemness and proliferation. And you see that, again, we have nice reversal of the phenotype. But it also brings up the question of what is really driving the memory of that phenotype after we've removed the arsenic. And so in these cells, we actually thought, well, memory's got to be epigenetics, or there must be some connection with epigenetics. And we used the XJB to ask whether the epigenetics is being driven by the arsenic or by the mitochondria. So complex figure, but essentially what we're looking at is the expression of the DMT3A methyltransferase. So at the RNA level, this is because one of the major drivers of epigenetics is methylation, and we had seen that arsenic in the muscles will give a good 2 to 3-fold induction of DMT3A. So in the C2C12s, same thing, arsenic will increase their DMT3A transcript. This correlates well with an opening of the promoter. So this is CHIP with Pol2 and looking at how open the promoter is. So Pol2 is being able to come in, and this is all reversed if we add the XJB. And again, reversed because we're adding it after we've removed the arsenic. This is looking at methylation of the DMT3 promoter, and you can see that the arsenic reduces methylation to increase expression, and somewhat reversed by the, actually that's the naive, this is the arsenic plus the compound, reversed by the compound. And interestingly, if we did CHIP with DMT3A on the DMT3A promoter, arsenic drives it off, and then you can see it returned to the promoter when we give the XJB. Another thing that came out was that we did CHIP with a number of different antibodies to different histone marks, and we could see different patterns of those mark changes with depending upon the gene that we were looking at. And some like H3K4 dimethylation really weren't affected, but something like H2AZ acetylation, which is usually seen in prime genes, was definitely very much affected. And again, this is another repressive mark that was reversed. And so we said, well, that's interesting. And we definitely had some muscles left over from mice that we had injected with XJB. And so we looked at this in vivo. And there was a little bit of a glitch in that in this experiment, 5 weeks of arsenic stopped the arsenic. And then we gave either vehicle or XJB to the mice with an IP injection for a week. Then we did our cardiotoxin injury, and then we looked at the muscles 2 weeks later. We looked at the TA muscles by histology, and the cardiotoxin is only injected into the TA. So we actually went for the biochemistry. We went to the gastrocnemius that wasn't injured, and looked at the DMT3A there. There was a vehicle effect on the arsenic induction of DMT3A, but strikingly, injection of the XJB really reversed the effects of the arsenic on DMT3A message, and then also on the marks that were going on to the onto the promoter to facilitate the induction of the DMT3A. So it brings up this hypothetical scheme that essentially says that, you know, when we have control muscles, we have healthy mitochondria, we have stem cells in the right place, we have connective tissue fibroblasts that are happy, we have a happy matrix. we injure these muscles, they go through a good regeneration process, and they will return to good, healthy muscles. With arsenic, we actually have a scenario where we set this up for bad regeneration by changing the mitochondria, changing the nature of the stem cells, changing the nature of the connective tissue fibroblasts, and so that we wind up with really aberrant repair overall. And so I've shown you what's going on with the mitochondria and the progenitor cells in all this, but what really was amazing to us was that there is a lot of memory that's out in this dysfunctional matrix that you see. I mean, that's a nice, healthy matrix, whereas this is discontinuous and not supportive. But I'll show you some evidence that there's communication of this memory even imprinted out into that matrix. And I'll also show that what we found was my old friend NF-kB was a major driver for a lot of this pathology. So again, returning to the those SHG images, in this case, we did an experiment where we had the mice on vehicle or a peptide that blocks NF-kB. It's a NEMO binding peptide that prevents NF-kB from being activated. Did this with our arsenic exposures as well. Again, took away the exposures and also took away the NEMO peptide. And then injured, and then in this case, we went out 4 weeks so that we could do the contractile testing. And looking 4 weeks after the injury, we see that, again, arsenic just does not look good for regeneration, whereas arsenic with this block of NF-kB, you see nice regeneration. So without NF-kB, arsenic didn't set the muscles up for bad regeneration. And we can quantify that looking at the volume of the fibers. But what was really amazing was that the matrix itself was changed in such a way that differentiation of the satellite cells into normal fibers was impaired. We found this out we took the gastric nemus out. We took all the cells out of the gastric nemus, so all we had was a cytoskeletal structure. And then we took that matrix, seeded human satellite cells on the matrix, and looked at the differentiation into Desmond positive myocytes or myoblasts in tubes versus fibrogenic cells. And we could do the imaging for green or red. We can quantify it, and as you see that we have an increase in cells that are differentiating to fibroblasts versus those that are going to muscle. And again, these are mouse matrices, but they're human myoblasts that were seeding on them, and those human cells never saw arsenic at all. And yes, we go back to the mitochondria. If we look at not the stem cells themselves, but looking at the connective tissue fibroblasts, we see that they also have these elongated mitochondria. And I'll point out that this is one kind of fused mitochondria, but if you trace all the way from here to here, that's one mitochondrion. It kind of goes in and out of phase, but you can see these large structures. These are those frustrated autophagy bodies that I mentioned as well. So we're seeing that in the connective tissue fibroblasts. Did a little bit of an array on those for expression of matrix proteins. And All the bars that you see in red are basically NF-kB driven transcripts. We also see that, again, there's those long changes in the mitochondria, and the what we see is we have that same change in the mitochondria. We can quantify length changes. We also looked at the abundance of different complexes in the respiratory chain. And we found that relative to complex 4, complex 3, and complex 5, that complex 2 and complex 1 were somewhat decreased. So again, some of that remodeling and changes in the mitochondrial respiration. But what was really interesting was that we could take the myofibroblasts out of the control or the arsenic mice. We could take them, plate them in a dish, allow them to elaborate extracellular matrix for 3 days, and then we took those cells away. All that was left was the matrix, and then looked at the effect on the human stem cell differentiation. And what we see is that the matrix that was elaborated by the arsenic-exposed connective tissue fibroblasts impaired the differentiation, seen here as Desmond, reduced MyoD, and increased the proliferative markers of CD34. And then also PDGFR alpha is expressed in fibroblasts that are tending towards adipogenesis. And I'll introduce you to another compound that we've used to test whether mitochondria are really involved with this. This is SS31. It differs from XJB in that it is not a radical scavenger. It is not an antioxidant per se, but the main idea of how it works is it actually protects the structure of cardiolipin in the respiratory chain. It improves respiratory chain protein interactions with cardiolipin and basically reduces ROS production by making the respiratory chain healthy. And we can see, looking at polarization of the mitochondrial membrane, again, it was increased by arsenic. This is in the C2C12 cells, but just like XJB, the SS31 was able to return things to normal. And again, we're adding the XJB after we took away the arsenic. In vivo, SS31 has a greater advantage because we can use saline as a control as opposed to the vehicle for XJB was DMSO that had that effect on arsenic. So no vehicle effect, but we saw that same type paradigm, arsenic exposure for 5 weeks, stop the arsenic exposure, inject saline or SS31 to the mice. Then in this case, we're using barium chloride, not cardiotoxin, for injury, basically an equivalent injury, and then going out 2 weeks. Control mice, arsenic mice, so again, bad repair. And in this case, if we gave the SS31, it reversed the effects of arsenic on regeneration, and we can quantify that. But I will point out one thing, that mitochondria, the SS31 definitely is mitochondrial active, and we've seen in many different endpoints that SS31 alone seems like it does some interesting things with either regeneration or mitochondrial action. But when we add arsenic and SS31 together, we definitely see a reversal of the negative arsenic effects. So this is a complex one again, but we took the connective tissue fibroblasts out of those mice that we exposed to SS31 or arsenic and SS31. These are the same ones you saw in the previous image, although that was an image of the TA. This is the gastrocnemius, where we took the fibroblasts from. And we put it through that same paradigm of plating the CTFs, letting them have time to elaborate a matrix, taking away the CTFs, and then plating the human progenitor cells on there. And what we see is that, again, we had some effect of SS31 alone, but here's arsenic and here's arsenic plus SS31. Now this one, I think it's an imaging artifact, because if you'll see, we have really beautiful structures with the SS31 alone, but they're thinner. And so I think it was just the way that we were doing the immunofluorescence, because clearly if you look at an arsenic exposure, you have aberrant structures overall, but they're returned to normal if we give the SS31. And the other interesting thing was that we could also reverse the epigenetics. I just want to go back for a second. So in this, we're collecting these cells 3 weeks after we took away the arsenic. So again, that idea of memory. And in this case, we're looking at DMT1 expression in those muscles 3 weeks after we've taken away arsenic, and basically 2 weeks after we stopped giving SS31. So without SS31, the muscles retained their expression, the increased expression of DMT1. This is mRNA. This is methylation of the DMT1 promoter. That's reversed by the SS31. And there's Pol2 again, getting great access with arsenic. And this is interesting, but it wasn't really the main explanation for why we were having effects with the DMT1, but DMT1 actually was still on the DMT1 promoter, which might indicate that it's not the major methylator in this one. But what was also really good was that we started thinking about, okay, so what would be modulating the phenotype of the cells, and especially what might be out in the matrix. And so one of the major cellular signalers that really dictates cell differentiation is the Notch system. And we looked at the epigenetics of Notch in the muscles, and we found that Notch was increased in expression with arsenic. That was decreased by the SS31. Pol2 had accessibility to the promoter, and that was reversed. And in this case, we went back to H2AZ acetylation, and you can see that permissive mark that was increased by arsenic is actually now decreased. So we have a reversal of the epigenetics because we added a compound that repaired the mitochondria. Looking at the myofibroblasts and their nature, arsenic gives you an increased activity of the Notch. This is the intercellular domain of Notch increasing with arsenic. SS31 reverses it. DLL4 is a Notch ligand, and the cells increase their expression of this Notch ligand, and that's reversed by the arsenic exposure. And we can go through a number of different areas here, but one of the things that we did was that, we showed this. This is actually what was going on in vivo expression, and these were cells that were taken from the different treated mice. Here we have the cells in the dish, and we're adding Dept to the cells. That's an inhibitor of Notch signaling, and we can see that we reverse the amount of intercellular domain, but we also reverse the ligand. So the ligand's really being driven by arsenic induction through Notch. And if we go with these cells and look at differentiation, in this experiment, we basically took those same cells, the arsenic-exposed cells versus controls, added the Dept to the arsenic, and you see nice differentiation of the human cells on that. Here we have the DLL4. What we did with the DLL4 was we allowed the cells to elaborate the matrix. We took away the connective tissue fibroblasts. Then we coated the matrix with an antibody that blocks the DLL4. Then we put the human cells on and looked at differentiation. And you can see that those cells differentiated really well compared to cells that were plated on the arsenic matrix. So back to our hypothetical scheme, essentially arsenic sets up a bad situation through NF-kB, through Notch signaling that changes the nature of the stem cells, the fibroblasts, imparts a memory into the matrix that's likely through Notch. But really the great thing is that if we give agents that reverse the effects of arsenic on the mitochondria, we can bring things back into normal health of the muscle fibers, health of the cells, and also regain our regenerative response. So just 2 more slides, 3 more slides to talk about translating this and bringing you back to the whole idea of the fat in the muscle, why that's important. This actually comes from interaction with and collaboration with Eva Milosevic at Pitt, and she's found that abdominal myostetosis, or that ectopic fat that I showed you, actually is an independent predictor of diabetes, of cardiovascular disease. We're looking into whether arsenic is associated with the myostetosis in some other studies, some epidemiology studies. My graduate student, Hung Bai, will be presenting this on March 16th in poster form, but basically looking at this idea of, does arsenic change the composition of the muscle fat overall? Bring you back to those pictures, and you can see that in this cellular infiltration after this injured muscle, a lot of this is fatty. Even in an uninjured muscle, we can see this increase in ectopic fat. But really, we're kind of wondering what the mechanism is there. And a lot of the mechanism, because this is interfiber, probably is changes in cell phenotype. We have this gain of the adipogenic PDGFR alpha. Many of the cells are pericytes, and pericytes are known to be able to be differentiated into adipocytes. And just quantifying all this and quantifying an increase in lipid in these muscles overall, we think that the main driver is a change in the cell phenotypes. And how's that happening? How's that being communicated across the fibers and different cells? We're heading towards looking at microRNAs that might be driving this. This is an array from a gastrocnemius muscle, and you can see that there's a large loss of microRNA expression in a number of different areas, fewer gains, but a lot of what is lost here actually are suppressing pathologies in the muscle. I'll point out 144 and 451. Very interested in those because they are major regulators of NF-kB activation. They suppress NF-kB activation. They're actually bicistronic, so it's nice to see that they both go the same direction. Here, we're actually looking at serum from the mice, and you can see that even in the serum, this is muscle tissue. This is serum. We can see that circulating levels of the NF-kB suppressing microRNA is gone. This is a muscle-specific microRNA that is very much involved in stopping the stem cell proliferation and advancing towards differentiation. And you can see that's also lost in the circulation. So last little scheme of where we are right now, we're trying to put all this together with microRNA changes affecting the NF-kB driven program that actually is driving epigenetic changes and bringing all the things together for how we actually have a memory in the tissues. And those are the folks who really do all the work and great collaborators. And I'll leave you with a picture of arsenic.
Thank you very much for very thorough and exciting talk and new areas in arsenic toxicology. For those who are still hanging around, we have some time we can take for questions.
I see a hand.
You there, or do you want me to read it?
Hi, Aaron. Great talk.
Hey, how are you doing, Bert?
Oh, I'm doing all right. So you opened up this topic with a question. Basically, will an environmental exposure prematurely age you? So my question to you is, do you have an aging reversal drug in either XJB or SS31?
So we actually used SS31 in a publication that was all on aging. While I was coming up with using the SS31, Fabi Ambrosio's lab that's really focused completely on aging and aging of those muscle progenitor cells was doing a paper and found that yes, the SS31 will reverse the effects of age on the stem cell mitochondria and their ability to differentiate and regenerate. So, yeah, it's kind of interesting. I think that there have been a number of people who have kind of looked at arsenic as maybe mimicking aging. I will say that there are many parallels, but arsenic always does things differently than anything else. So the SS31 is actually in clinical trials or getting through clinical trials. It's being used to improve cardiomyopathies, and the folks who have developed it have some really great studies looking at reversal of age in the heart and then also in skeletal muscle.
Jamie Young has a question in the chat. Is it possible that some of the impact that arsenic is having on the muscle is through its ability to depolarize tubulin?
Depolarized tubulin?
She's wondering if some of that impact is via that mechanism.
Sure. So there's a lot of effects of arsenic on cytoskeletal elements. We have seen, I want to say that was in liver. We have seen kind of disruption of the tubulin changes. We've seen major changes in matrix overall. And a lot of that probably has to do with arsenic effects on GTPases. Some of those mitochondrial enzymes that I showed you changing, their GTPases. We've shown that arsenic will give you a chronic elevation of RAC activity. Yes, quite possible.
Okay, Mike Banerjee and then Wenky Phone.
Oh, hi, this is 1K. I have maybe a naive question that we know the muscle loss or sarcopenia, that's one of the characteristics is that branch-chain amino acid loss. And we know the branch-chain amino acid loss is known to strongly stimulate mitochondrial biogenesis in aging subject. So my question is that arsenic exposure change in metabolism that leads to the branch-chain amino acid loss, decrease. Possibly we can just connect to broad question, possibly we can use branch-chain amino acid to attenuate arsenic exposure-induced muscle loss.
I think it's a pretty good idea. I think it's quite plausible. We seem to have a lot of effects on complex 1, complex 2 in the respiratory chain. We've actually focused a lot more on beta oxidation changes and lipid, because we're more focused on lipid changes. But I'm sure that there are probably effects with the branch chain amino acid synthesis as well.
Mike Banerjee.
That was a great talk, Dr. Barchowsky. So I was wondering, you have used 100 ppb, if I remember, for mice, which is on the lower end of the spectrum, and we still see all these effects. So mice are known to have better clearance of arsenic than humans. So do you have any idea what was the dosage at the tissues, and how does it compare to chronically exposed humans? Because we might actually be at risk from even lower dose than what we think we are.
I don't know how many years I've had this conversation with Dr. States, and even published with Dr. States on this. But you're absolutely right. And so we've actually published dose response for arsenic changes in the vasculature and tissue remodeling. And we can see changes even at 10 parts per billion in the mice. That said, if you go through all the calculations of human equivalent dose, it's scary that we're probably looking at maybe a human equivalent of about 20 parts per billion exposure. So that says that arsenic, the mice might be a lot more sensitive for tissue remodeling than humans. Or we just don't see it in humans as quickly as we can see it in mice. But I think that one of the things that we've definitely shown over the years is that even though they eliminate it quicker, they can have effects on cellular receptors. So you could still have, you don't have to take the compound into the tissue necessarily to get the effect. And then also, I would say that if you went with humans and the epidemiology, basically if a human's drinking about 100 parts per billion, they're going to have a blood arsenic level of maybe 90 nanomolar. And then if you go to tissue distribution, our use of 20 nanomolar in the cell culture is kind of in the range that you might see actually in the skeletal muscle itself.
Okay, James Wise has a question in the chat. Do you think the results are unique to arsenic, or could it occur with exposure to another metal like chromium, nickel, or cadmium?
Well, James, you should know that chromium is not distributed in the body like arsenic. So I would say it's probably a moot point, although there were some similarities with chromium and arsenic when we were looking at lung epithelial cells, but actually chromium opposed some of the actions of arsenic in those mice, and also in the lung cells. So arsenic has a different profile of disease, and even differences in cancers, although that's probably a kinetic thing. But no, they're definitely different. There are some similarities with nickel, but arsenic, I think, is very unique overall in its ability to bind to thiols and give cellular reactions. In saying that, Matt Rand up at Rochester is doing very nice parallel studies to ours using mercury. So the thiol reactivity of mercury might be a little bit more of a similarity there.
And Jamie Young's got her hand up.
Amy? I do. I can ask in person as much as it can be this time. But so my question is kind of looking at the difference again between males and females, because I noticed that the cell line that you're using is actually from a female mouse. And so my question, there's a couple of them. One is, why did you choose to use a mouse cell line for the myoblast and not a human cell line? And then if you did this in males and females respectively, do you think you would see a difference in not only your outcome with arsenic, but how those drugs are working? Because there's difference in energetics between muscles. So I'm just curious as what you think, where it's important and where you think the outcomes might be different.
Okay. I actually showed you some female data, but I didn't point it out. But you're absolutely right. There are differences between males and females. Why did we use C2C12s instead of human cells? Because C2C12s grow like weeds, and you can do a lot of experiments to get you in the right ballpark to then spend a lot of money to do human cell work. So, that's a cheap way out, but what we always came back to was trying to show that cells from in vivo mouse experiments also were effective. We have done some work with the human cells in culture as well. But to the sex difference, we definitely have differences in some parameters with the arsenic and some not really. We saw a large difference between males and females with adiposity, and regenerative wise, we don't really see that much difference. And what I can say is that we inherently do male-female comparisons because we often do in utero exposures, and you get 50-50 pups, and looking at those, it's really hard to tease out a lot of differences in the regenerative response between the sexes, but some of the gene expression and I think some of the microRNA expression, if I remember correctly, is a little bit different. So there definitely are differences there.
Okay, Wenky, did you have another question, or was your hand just still up?
No, I have no question.
Banrita Walam just raised her hand. Banrita?
Okay, I have to block that hand. Okay.
Hello, can you hear me?
Yeah.
Yes.
Okay, so I have more of a public health question rather than on your experiment. So I'm just wondering, when you've been working in arsenic for a long time, like you said, so when you were looking at these cardiovascular and metabolic outcomes in human populations, did you see a difference between, like, let's say countries like Pakistan and India versus more developed countries? Like, were these outcomes identified sooner, or were they, let's say in Pakistan and India, were they identified at a much later stage of the disease?
That's a very good question. And I'll say that one response is that Bangladesh and Taiwan really gave us a lot of our appreciation that arsenic in the environment causes disease. And one of the striking things that you saw there was that there was really high exposures that cause skin lesions. And so you could say, whoa, I see skin lesions on the hands. But, and some people say that's somewhat unique to those countries because you don't really see skin lesions in the US or something, even though there's equivalent doses. But it's really getting an appreciation for that, and then getting an appreciation for the number of other cancers that were driven by arsenic in the various populations that were being looked at, that then you went to more subtle things like non-cancer diseases. And when people really started looking for it or measuring arsenic in urine, et cetera, they realized, well, this is a lot more pervasive than you'd expect. There's a lot of bias that there is no arsenic disease in the United States, which Dr. Stace is shaking his head, but we heard that a lot with our committee work. And one bias is that people in the United States don't drink water, but they die of high fructose corn syrup. But the real upshot is that it's not until people really looked for the exposure that they really realized that arsenic was a major driver.
Yeah, actually, talking to dermatologists, I learned that there are skin lesions in the US. They're just not reported.
Yeah.
So, okay, we're a bit over, but it's been fascinating. Great talk, Aaron. And thank you all for attending. I think we have another meeting on another link.
Okay, so I'll leave and then join.
Yeah, see you in 5 minutes.